Brucellosis. It's a bacterial infection, kind of stealthy, lives inside cells.
Yeah, it ducks the immune system, can cause these really debilitating symptoms that flare up.
And it's also quietly devastating animal populations, um, economies too. So that's what we're diving into today.
Exactly. And what's, I think, really compelling about brucellosis is just how different its impact can be, on human health, on agriculture. It really depends on the animal source.
That's right. So our mission here in this deep dive is to unpack some source material that compares two, well, wildly different animal sources for this disease, camels and deer.
We want to give you a shortcut sort of to understanding the very distinct challenges and the tailored solutions that each one needs.
So why should you care? What's the relevance here?
Well, brucellosis, it's definitely more than just another global health issue. It's a fantastic case study, really, in this one health idea.
Right, the interconnectedness.
Exactly, how human, animal, and environmental health are all tied together. And it really highlights the huge economic damage it can cause.
From individual farms losing productivity.
All the way up to international trade restrictions that can, hit entire countries. It's a genuinely global, multi-layered problem.
Okay, let's unpack this thing. Brucellosis. It's a bacterial zoonosis, meaning it jumps from animals.
And it's sometimes called undulant fever or Malta fever. That undulant part refers to the fever pattern.
Yeah, that characteristic rising and falling fever, sometimes over weeks. It's found pretty much globally.
And the estimates for new human cases each year are just staggering, somewhere between 500,000 and potentially over 2.1 million.
Wow. And the WHO calls it a neglected zoonotic disease, right?
Mm-hmm.
Especially hitting lower income nations.
That's where you often see people living really closely with their livestock, so the risk is higher. And this immediately brings us back to that one health idea being so critical.
Because human infections almost always come from animals.
Precisely. So if you want to control it in people, you have to control it in the animal reservoirs. It's non-negotiable.
Meaning you need public health, vets, wildlife managers, everyone working together.
You absolutely do. You can't tackle one piece in isolation. It just doesn't work.
Okay, so the agent itself, the Brucella bacteria,
Yeah.
what's the key thing we need to know about it?
Well, biologically, the really critical feature is its knack for surviving inside host cells. This kind of stealth mode is what makes it so persistent.
And so hard to treat.
Exactly. Now, there are a few key species to be aware of. Brucella melitensis, that's usually seen as the most dangerous for humans. You often get it from goats, sheep, and camels.
Okay.
Then there's B. abortus. Think cattle, bison, elk, but also camels and deer can carry it.
Right.
B. suis is linked to pigs, but also caribou, reindeer, and deer again. And B. canis from dogs, which generally causes a milder illness in people.
So back to that stealth mode, surviving inside cells, especially macrophages.
Yes, that's the central trick Brucella uses. It basically hides out from the immune system, finds a little protected spot to multiply.
And that intracellular life is why it causes chronic problems.
It is, and why treatment is such a challenge. You need antibiotics for a long time, like 6 weeks or more, and usually a combination of drugs that can actually get inside those cells.
And even then, relapse can happen.
Relapse is definitely a significant worry, yeah.
Okay, so how does it work in animals? How do they get infected and spread it?
Typically, they ingest contaminated stuff, feed, water, that kind of thing, or it gets in through mucous membranes.
And it likes certain parts of the body.
It has a strong preference, a tropism for reproductive organs, and also the reticuloendothelial system, lymph nodes, spleen, liver.
And I read about this sweet spot for its growth. Erythritol.
Ah, yes, erythritol. It's a sugar alcohol found in high amounts in the placenta and fetal fluids of many animals. Brucella loves it, basically. It helps it grow rapidly there.
Which leads to the reproductive problems.
Exactly. Abortions, stillbirths, infertility, sometimes reduced milk or meat yields. That's the big economic hit for farmers.
And how does it then spread to other animals?
The main way is after an abortion or even a seemingly normal birth in an infected animal. There's just a massive shedding of bacteria in the uterine fluids, the placenta, the fetus itself.
Contaminating the whole environment.
Heavily contaminating it. That becomes the primary source for others in the herd.
And for humans, we're just accidental hosts.
Pretty much. We get it through direct contact with infected animals, or often by consuming contaminated animal products.
Like unpasteurized milk or cheese.
Precisely. Symptoms in humans can be tricky because they're often nonspecific. Flu-like stuff initially, fever, those drenching sweats, feeling unwell, muscle and joint pain.
But that undulant fever is a bit of a giveaway sometimes.
It can be, yeah, that pattern of fever rising and falling.
And if it's not treated, it can get chronic,
Oh, absolutely. It can lead to some really debilitating long-term complications, affecting almost any organ system, really.
like arthritis.
Arthritis, spondylitis, inflammation of the spine, endocarditis, inflammation of the heart lining is actually the most common cause of death from brucellosis. Neurological problems can happen too.
But crucially, it doesn't usually spread person to person.
That's a really important point, yes. Human-to-human transmission is exceptionally rare. So it's devastating for the individual, but not typically a public health spreading crisis in that sense.
Okay, let's do that first deep dive then. Brucellosis from camels. You said this is a severe escalating threat.
It really is, particularly in pastoralist communities across Africa, the Middle East, and Asia. It's deeply woven into the social, cultural, and economic life there.
And camels can get multiple brucella types.
Yeah, they're susceptible to B. melitensis, the more human pathogenic one, often picked up from sheep and goats they might graze with, and also B. abortus, typically from cattle contact. There's even been B. sus DNA found.
And it's widespread geographically.
Pretty much everywhere camels are raised, except Australia, apparently. That global meta-analysis put the average prevalence around 9%, but that hides massive differences.
Like Sudan versus Oman.
Exactly. Sudan, you might see herd prevalence up to 40%. Oman, maybe less than 0.5%. A lot of this spread is driven by uncontrolled animal trade and movement. Animals moving across borders without checks.
How does the way they're farmed affect things? The husbandry.
There's a really clear difference. Nomadic herds moving over large areas tend to have low prevalence, maybe 2–5%.
Okay.
But when you get into more intensive or semi-intensive systems where animals are more crowded, maybe mixing with other species,
Prevalence shoots up.
yeah, 8–15%, sometimes even higher. Co-rearing with sheep, goats, cattle is a big risk factor. Larger herd sizes, higher density, and introducing new animals without quarantine, these open replacement systems.
So if it's spreading in these herds, how easy is it to spot? You mentioned silent carriers.
That's a huge challenge. Infected camels often show few or no clinical signs. They look perfectly healthy, but they can be shedding the bacteria.
So not obvious signs like in cattle sometimes.
When signs do appear, they are mostly reproductive abortion, stillbirth, inflammation in males. But interestingly, retained placenta, which is common in infected cattle, seems rare in camels.
And diagnosing it is tough too.
Very tough. The standard blood tests, serological tests, were mostly developed for cattle. They often just aren't accurate in camels unless they've been specifically validated.
So you need multiple tests.
Yeah, experts usually recommend using a combination of tests, like the Rose-Bengal plate test plus an ELISA, to get a more reliable picture.
Okay, silent carriers, tricky diagnosis. So how does this become such a human health crisis in these areas? How are people getting it?
The number one route, overwhelmingly, is foodborne.
The raw milk again.
Raw, unpasteurized camel milk and dairy products made from it, cheese, buttermilk, things like that. And this isn't just occasional. It's often a deeply ingrained cultural practice.
People believe boiling it ruins it.
Yeah, there's often a belief that boiling reduces the nutritional value or spoils the taste. So this creates a really direct, continuous exposure pathway for the general population, women, children, everyone.
And there have been outbreaks linked directly to the milk.
Oh, yes, definitely. Clear outbreaks trace back to contaminated camel milk.
What about other routes? Direct contact.
That's the secondary route, yeah. Mainly occupational, people handling camels, daily milkers, herders, vets, family members helping with births. They're exposed to infectious birth materials, blood, etc.
And is there awareness of this risk?
Often very little. Many pastoralists just aren't aware that brucellosis can jump from their animals to them. So practices like handling aborted fetuses or placentas with bare hands are unfortunately common.
So you've got the raw milk consumption, the direct contact, the lack of awareness.
It really is a perfect storm for endemic human brucellosis. High exposure risk meeting low awareness.
Okay, let's pivot completely now. Brucellosis in deer. You said this is a different picture, especially in places like the US and Canada.
Yeah, in developed countries where brucellosis in livestock, like cattle, has largely been eradicated, deer present a different kind of challenge. It's more about the wildlife-agriculture interface and maybe some occupational risk.
And different Brucella species involved too.
Generally, yes. Various deer species, white-tailed deer, mule deer, elk, can get infected, but it's usually not B. melitensis like in the camel situation.
It's the ones linked to cattle or pigs.
Mostly B. abortus, which they can pick up from infected cattle or often from wild reservoirs like bison or elk, or B. sus, which they can get from feral swine populations.
So the big question is,
Yeah.
are deer a self-sustaining reservoir for this? Or are they just catching it occasionally?
The evidence really points towards the latter. They're more likely incidental spillover hosts in most areas.
How do we know that?
Well, numerous surveys, particularly in North America, show very low prevalence in white-tailed deer populations. Like that Missouri study you mentioned, one positive out of over 700 deer.
Which is very different from known reservoirs.
Exactly. Contrast that with bison and elk in the greater Yellowstone area, or feral swine, which are established, persistent reservoirs that maintain the bacteria. Deer don't seem to do that in the same way.
So if they're not really a reservoir, what's their significance? Why are we talking about them?
Their main role seems to be less about causing widespread human disease directly and more about acting as a potential bridge or conduit
A bridge between what?
between the primary wildlife reservoirs like elk and brucellosis-free domestic cattle. If a deer gets infected from elk and then interacts with cattle, it could potentially transmit it.
Jeopardizing that brucellosis-free status for livestock.
Precisely. And that's a huge economic threat to the multi-billion-dollar cattle industry. So it really reframes the deer issue. It's less a direct public health crisis, more an economic and ecological management challenge.
And clinically, are infected deer obviously sick? Like the camels, often not.
Similar story, yeah. Infected deer often show no visible signs of disease. They look healthy. When signs do occur, again, typically reproductive, like abortion.
Which means hunters can't tell if an animal is infected just by looking at it.
Nope. No way to tell visually.
So what about the human risk from deer? Who's at risk and how?
It's a much more specific risk profile. Primarily hunters, wildlife biologists, maybe veterinarians who handle deer carcasses or samples.
And how are they getting exposed? Not the milk route, obviously.
Right, it's not foodborne like the camel milk situation. It's almost entirely through direct contact with infectious materials during field dressing, butchering, handling the carcass.
Bacteria getting into cuts or maybe mucous membranes.
Exactly. There's also a potential risk from eating undercooked venison, but
the actual risk seems low.
it appears quite low. Even though we can detect B. abortus antibodies in deer sometimes, there aren't really definitive reports of people getting brucellosis from deer. It seems more theoretical than a common occurrence.
Okay, this is where it gets really interesting. The aha moment, maybe. Comparing camels and deer side by side.
It really crystallizes things, doesn't it? The zoonotic threats from these two animals operate in fundamentally different ways, different paradigms entirely.
Meaning a one-size-fits-all control strategy is just not going to work.
Completely inadequate. The camel threat is what you might call a source problem.
Meaning the camel population itself is the main generator.
Yes, it's a continuous high-output reservoir, mainly affecting a broad human population through that foodborne route, the milk.
Whereas the deer threat is more of a conduit problem.
Exactly. Deer act more like a potential bridge, transferring the infection between other reservoirs like elk or swine and maybe occasionally to livestock, or posing a direct contact risk to a very specific limited human group.
And getting this distinction wrong is dangerous.
It's critical. Imagine trying to apply hunter safety advice, wear gloves when butchering in a pastoralist community where the real risk is drinking raw milk daily. You'd completely miss the mark.
Or pushing for milk pasteurization in a region where the only brucellosis link is deer hunting. Pointless.
Totally irrelevant. And you see this clearly when you compare the key features.
Okay, let's run through those. Primary Brucella species.
Mostly B. melitensis in camels, which is highly pathogenic to humans, versus B. abortus or B. sus in deer, which are moderately pathogenic.
Geographic focus.
Endemic, widespread problem in developing nations for camels. Sporadic, localized issue, mainly wildlife interface, in developed nations for deer.
Animal host role.
Camels are a significant maintenance host. Deer are likely just spillover or conduit hosts.
Primary human transmission route.
Foodborne raw milk for camels. Direct contact during carcass handling for deer.
Main human population at risk.
The general population, especially women and children, in pastoralist areas for camels. Specific occupational or recreational groups like hunters for deer.
And the main driver for control efforts.
Public health crisis for camels. Protecting the national livestock industry for deer. It's just Fundamentally different.
Wow. Okay, so given these completely different scenarios, the control strategies have to be tailored. You need a specific toolbox for each situation.
Absolutely. A holistic strategy for camels won't work for deer and vice versa.
So for camels and the pastoralist communities, what's in that toolbox?
It has to be multi-pronged. On the animal health side, you need to reduce the infection in the camels themselves. Vaccination is key.
Using existing cattle vaccines.
Often, yes, things like B. abortus S19 or B. melitensis reveron 1 are used. But honestly, there's a real need for vaccines and diagnostics specifically developed and validated for camels. That's a gap.
Test and slaughter could be an option too.
Maybe in some high prevalence, more controlled settings, but it's often difficult in vast pastoralist systems.
What about biosecurity? Simple farm practices.
Yeah, things like trying to keep camels separate from potentially infected sheep, goats, or cattle. Better hygiene around births. Quarantining any new animals brought into the herd. Basic stuff, but important.
And then the public health side, breaking that milk transmission.
That is absolutely critical. Public awareness campaigns are essential. Hammering home the risks of raw milk and strongly promoting boiling it before drinking.
But that needs to be done carefully. Culturally sensitive.
Hugely important. You have to respect traditions and beliefs about milk while clearly explaining the health risks. It's a fine line.
And involving the community itself.
Essential. Top-down approaches rarely work well. You need a participatory approach, working with community leaders, herders, education on practical things they can do.
Like wearing gloves for births, safe disposal of afterbirth.
Exactly. Simple, practical steps to reduce direct contact exposure.
Okay, now shifting to the deer side. Managing that wildlife-livestock interface and the hunter risk. What's the strategy there?
It's much more targeted. First, wildlife surveillance. Continuously monitoring deer populations for brucella, especially in areas where they overlap with known infected wildlife, like
Right.
the Yellowstone area, or with livestock farms.
As an early warning system.
Precisely. Then, for hunters, the main human group at risk, it's all about education and prevention.
Getting the message out through hunting licenses, websites.
Yes, targeted communication. Emphasizing wearing PPE, rubber gloves, maybe eye protection when field dressing and butchering.
Minimizing contact with certain parts.
Yeah, especially reproductive organs or fluids. Thorough hand washing afterwards is crucial. And this is really important, cooking venison thoroughly.
What temperature?
At least 165 degrees Fahrenheit or 74 Celsius. And reminding people that freezing, smoking, or drying the meat does not kill the bacteria.
Cooking is key. What about disposing of the carcass?
Proper disposal is important too, to prevent scavengers from potentially picking it up and spreading it further.
And on the livestock side, protecting the cattle.
That's the big economic driver here, remember? So robust biosecurity on farms is key. Trying to minimize contact between cattle and deer, especially during calving season when cattle are most vulnerable.
And continued cattle vaccination in risky areas.
Yes, vaccination remains a critical tool for livestock protection in those interface zones.
And what about the professionals, vets, lab workers?
Strict occupational safety. Using appropriate PPE when handling animals or samples. And any suspect Brucella samples need to be handled under Biosafety Level 3, BSL-3, conditions in the lab.
Okay, so wrapping this all up. The big takeaway seems to be that brucellosis isn't just one single problem.
Not at all. It's a mosaic, really, of distinct challenges, and the camel versus deer comparison illustrates that perfectly.
One is a widespread public health crisis tied into culture and economy.
The other is primarily an agricultural and wildlife management issue with a much narrower specific human risk profile.
So the path forward, what are the key recommendations?
Well, it underscores the need for more investment in real One Health approaches in those endemic camel regions programs that are culturally smart and involve the community.
And for the deer situation.
Targeted risk communication for hunters and vets. Robust surveillance where wildlife, livestock, and humans interact.
And research.
Definitely. Prioritizing research for better tools for camels, diagnostics, vaccines, and understanding those transmission dynamics better, especially at the wildlife-livestock interface.
Which leaves us with a final thought for you, the listener.
Yeah, something to chew on. How much might our understanding of any zoonotic disease be limited if we don't first stop and really ask? Are we dealing with a source problem, like the camels? Or a conduit problem, like the deer? Or maybe something else entirely?
How many other one-size-fits-all solutions out there might be missing the mark because we haven't asked that fundamental question first?